Welcome, science friends, to Gregor Mendel and his work with genetics. Gregor Mendel was born a long time ago in the early 1800s, and he was Czech. He studied science and math at the University of Vienna and spent a long time teaching and working in a monastery. He did his most important work at that monastery in the garden. Gregor Mendel is known and very famous for working with pea plants when he was studying genetics. Mendel's work was based on pea plants. He cross-pollinated what we call true breeding parents. So true breeding means that both parents were homozygous. He raised them from seeds and observed traits. This allowed the offspring to self-pollinate, meaning one generation of flowers pollinated with themselves. And then he continued to observe what happened with next generations. When we're looking at how traits are passed down through generations, we need to be able to tell the difference between each generation. So we need to name them. The very first generation is labeled with a P, P for parental, or the parent plants, or the parent generation. The offspring, or the children of that parent generation, is known as the F1. F stands for filial, it's just old Latin stuff, but basically the 1 tells us it's the first generation after the parents. The generation after that is F2, it's the second generation after the parents, and it keeps going and going, F3, F4, F5, F6, all the way down. Let's take a closer look at Mendel's work. So here we have our parent generation. We have true breeding purple flowers and true breeding white flowers. And again, true breeding means they have a homozygous genotype. So both of their letters are going to be the same capitalization. What Mendel saw next was the F1 generation gave us 100% purple flowers. So this was an interesting experiment. We start with two different color flowers, and when we breed them together, we only get one color. So in that F1, or first offspring generation, we saw 100% of those flowers be purple. So Mendel had to think to himself, where did those white flowers go? They didn't just disappear. That couldn't be possible. Otherwise, we wouldn't get white flowers in the future. So what he did is he took that F1 generation and he bred them together, or he self-pollinated. So all of the flowers on that F1 generation, he bred them together. And that resulted in the F2 generation, where we saw 75% of the flowers were purple, and now 25%, or a quarter, were back to white flowers. Interesting. It seems like those white flowers skipped a generation. They came back. Based on this experiment, Mendel discovered that the purple flower allele is dominant over the white flower allele. Because purple is the dominant allele, we're going to use P as our letter to represent our alleles. So when he bred those two true breeding plants together, the all purple, all white, again, this is what he found. If we have purple is dominant and they're true breeding, that means they're homozygous, so the same size letter. The purple parent was big P, and the white parent was little p, because the white is recessive. It needs two little letters. And when he bred those together, we got one from each. We got a big P from the purple plant and a little p from the white plant. So all of that F1 generation is heterozygous, one big, one little. When we're looking at how to use our vocab this unit, the phenotype is the color of our flower. Is it purple? Is it white? The genotype are the letters. Which P's are we using? Are they big P's, little p's, or a combination? So here we see the F1 generation. Again, 100% were purple. Well, why is that? Because, again, we're getting a big P from one plant, a little p from the other plant. So all of the F1 generation is heterozygous. They get one big, one little. And according to the laws of genetics, if there is a dominant big letter in a genotype, that trait will be expressed. And since there's a capital P for all of our genotypes, they're going to be the purple color. Now, what's going on with that F2 generation? So let's take these heterozygous purple flowers and breed them together. We're going to self-pollinate. And what that results in is our 3-to-1 ratio. So 3 out of the 4, or 3/4 of them, will be purple. 1 out of 4, or 25%, will be white. And how do we know that? We have to look at those genotypes. So here's where genotypes and phenotypes start to get confusing. Two organisms can have the same phenotype, so the physical representation, in this case the color. We can have two organisms with the same purple color flower, but they can have different genotypes. The letters can be different. Even though we have the different genotype, we can have the same purple phenotype. Because we can have different amounts of phenotypes and genotypes, let's see what that means for our F2 generation of Mendel's flowers. A quarter, or 25% of the F2 generation, is going to be homozygous dominant, big P, big P. Half, or 50%, will be heterozygous, big P, little p. And the last 25% will be homozygous recessive, little p, little p. So our genotype ratios are 25%, 50%, and 25%. But our phenotype, we see 75% are the color purple, because we have a big P present, and that dominant purple gene will be expressed or shown. And 25% will be white. So our genotypes and phenotypes can be different ratios. The letters versus the colors. We can have different amounts of genotypes and phenotypes. They aren't always going to be exactly the same amounts. Sometimes they are, but not always. That was Mendel and his work. I hope it helped.